In this topic, you must know various definitions. What are determinants of antigenicity? And what is HLA complex? We can define antigen as a substance which, when introduced parenterally into a body, evokes an immune response. It produces specific antibody or specialized sensitized T lymphocytes or both. And in this definition, what is important is parenterally. Parenterally means other than enteric route. Parenterally means other than enteric route. Usually the antibodies which enter through oral route, they are destroyed by digestive enzymes and no antibody is produced. Exception to this is vaccines, oral vaccines which can be given orally. Some vaccines they can produce antibodies after oral administration. So in definition, what is important is parenterally, second is specific. So antigen will react with its specific antibody and no other antibody. Antigen will react with only those lymphocytes that are having receptors for these antigens. So this is very important term used in the definition, specific. And antigen reacts with the antibody in observable manner. What is immunogen? It is a protein, carbohydrate that can initiate a detectable immune response. Ability of an antigen to induce immune response is called as immunogenicity, whereas ability of an antigen to combine specifically with the antibodies or T lymphocyte receptors is called as antigenicity. Another term which is frequently used is hapten. What is hapten? These are low molecular weight substances which are unable to induce antibody formation by themselves but can produce antibodies when they are linked to a carrier molecule. They can react specifically with the antibody produced, and examples of haptens are penicillin, picril fluoride, and formaldehyde. You can see in this photograph hapten when it is bound to a carrier molecule like albumin, globulin, or synthetic peptide. It can produce antibodies and react with the antibodies. In this case, antibodies are produced not only against hapten but also against the carrier molecule. What are types of hapten? Hapten can be simple hapten or complex hapten. Simple hapten, they are low molecular weight substances which are usually univalent. They will combine with the antibody but no precipitate is formed. But they can inhibit precipitation by corresponding antigen or complex hapten. Whereas these complex haptens, they are large molecular weight substances which are polyvalent and they combine with the specific antibody to form a precipitate. Example of complex haptens are capsular polysaccharides, pneumococci, and cardiolipin. Simple hapten example is penicillin. And what is about heterophile antigen? What are these antigens? Certain antigens, they are similar in nature. They are not identical, they are similar in nature. They are present in different tissues of more than one species. Examples of heterophile antigens are Forssman antigen, cross-reacting microbial antigens which can be seen in Rickettsia and Proteus, which are used in Well-Felix reaction, RBCs of blood group B, and antigens of E. coli. These are heterophile antigens. So antibodies produced against one antigen can react with that other antigen, cross-reacting antigens. Forssman antigens, they are distributed in different cells. So they can be seen in red cells of horse, cat, mouse, sheep, and chicken, kidney cells of guinea pig, and various bacteria. When tissue from guinea pig is injected into rabbit, it will produce antibodies against RBCs of sheep. So this is because of similar nature of antigen. So antigen is shared between different tissues of different species. Another term which is used commonly is pro-antigen. These are low molecular weight substances that will not produce antibodies, but they can cause delayed type of hypersensitivity reaction. Dinitrochlorobenzene is an example of pro-antigen. Epitopes. Epitopes, they are present on antigens. They are smallest unit of antigenicity, made up of few amino acids. Their size measures about 25 to 34 angstrom unit and about 400 to 1000 molecular weight. These epitopes, they are present on antigen and corresponding area on the antibody, it is called as paratope. T lymphocyte usually recognize sequential epitopes where the antigenic determinant is present in a linear form or sequential form. Where B cell recognize conformational epitopes. What are conformational epitopes? They are formed by the surface residues at different sites of peptide chain. By folding, they are brought together by folding to form a tertiary structure. And B lymphocytes can recognize these conformational epitopes. And paratope is a combining site on the antibody molecule to which epitope attaches. What is valency? Valency is a number of antigenic determinants on the surface of an antigen. Multivalent antigen induce stronger immune response than the monovalent antigens. Valency can be functional valency, where the number of antigenic determinants they are included in functional valency, and in non-functional valency, antigenic determinants are present on unexposed region of antigen, they are included. So total valency of antigen, it includes functional valency as well as non-functional valency. You can see this is an antigen molecule. If you see, there are 4 sites or antigenic determinants present on this antigen. You can see this is functional valency is 4. Another example where you can see antigen with 3 antigenic determinants. So functional valency of this antigen molecule is 3. So this is the smallest unit of antigen to which antibody binds. This is called as epitope, and paratope is the area, corresponding area on the antibody molecule. What are determinants of antigenicity? There are different factors that will determine antigenicity of a substance. First is foreignness. Normally, there is no immune response produced against self-antigens, and antigenicity is directly proportional to the degree of non-self nature or foreignness. More foreign is antigen, more is the immune response produced. That's why there are plant antigens, they will produce more immune response as compared to animal antigens. Large molecules with molecular weight more than 10,000, they are highly antigenic. Molecular weight less than 5,000, usually these substances, they are non-antigenic, and those substances with molecular weight of 5,000 to 10,000, they are less antigenic. We can convert these non-antigenic substances into antigenic substances by absorbing on inert particles like bentonite particles or kaolic particles. Picril chloride, formaldehyde, penicillin may be antigenic when they bind to a carrier molecule. So second factor that will determine antigenicity of a substance is the size of molecule. Third is structural diversity or chemical nature. Most of the antigens, they are proteins. The proteins which are having 20 amino acids, they are better antigens as compared to polysaccharides which are having 4 to 5 monosaccharides. Exception to protein antigen is gelatin. Usually aromatic radical is essential for antigenicity. Lipids and nucleic acids, they are less antigenic as compared to proteins. Other factor which can affect susceptibility to tissue enzymes. Substances which are metabolized and susceptible to enzymes, they behave as antigens. Substances like synthetic polypeptides which are not metabolized, or substances like latex particles which are not susceptible to enzymes, they are non-antigenic. When a molecule, antigen molecule is broken down, its hidden antigenic determinants, they are exposed. So, for example, bovine serum albumin is having 6 antigenic determinants on the surface. When it is broken down, there are more than 18 antigenic determinants. So after treating with the enzymes or after metabolism, more antigenic determinants are exposed. So the antibodies produced or the immune response is more when the molecule is broken. Next factor is genetic factors. There is difference in the immune response to same antigen because of genetic factors. Some individuals, they are good responders where the antibody production is fast. Some are slow responders. Some are non-responder where no antibody production can be seen. Dose of antigen. If the dose is too little, it will fail to produce immune response. If it is large, it can lead to tolerance, development of tolerance. So repeated doses, they are required for production of adequate immune response. Next factor that will affect is route of administration. Oral antigens, usually they produce IgA type of immunoglobulin. Colon antigens, when they are inhaled, they will produce IgE type of immunoglobulin. So route of administration will also determine what type of response is seen. Site of injection is also important. Hepatitis vaccine is more immunogenic when it is given on deltoid as compared to injection given on gluteal region. Other factors which can affect are if the antibodies are given prior to administration of antigen, then immune response is suppressed. These are the factors that will determine antigenicity of a substance. Now, what is antigenic specificity? Position of antigenic determinant in a molecule will determine antigenic specificity. So if the antigenic determinant position is changed, the antigenic specificity is changed. Also, when there is change in the configuration of antigenic determinant, isomers, dextro, livo, or meso isomers, they will show different type of antigenic specificity. All individuals in a species, they possess species-specific antigen. Examples are human blood proteins or animal proteins, and they can be helpful in tracing evolutionary relationship between species or identification of species from blood and seminal fluid. Isoantigens are found in some, not all the members of the species. Example of isoantigens, they are blood group antigens, which are important in blood transfusion. They are important in isoimmunization during pregnancy. What is isoimmunization? In pregnancy, if mother is Rh negative and father and fetus is Rh positive, in first delivery, some fetal cells, they are transferred to maternal circulation and antibodies are produced against these Rh positive cells. So this is called as isoimmunization. So these antigens, they are present in some members of a species, not all the members. Therefore, they are called as isoantigens. They are also important in evidence for disputed paternity. Actually, antigens which are important for transplantation, they are also examples of isoantigens. They are found in some species, they are not present in all the species. So they are present in some members of a species, not all the members. Self-antigens, they are non-antigenic. Some antigens which are hidden antigens like lens proteins, which is covered by capsule, is a sequestered antigen. Sperms are produced at the time of puberty. So immune system is not exposed to these hidden antigens. When these antigens, they are modified either by infection or by irradiation, they can lead to immune response. Organ specificity. Antigens between different organs, they are shared. For example, organs like brain, kidney, or lens protein, the antigens of different species, they are shared. Because of sharing of antigen, for example, in case of brain specific antigens, they are shared by the man and sheep. when the sheep brain is used for vaccine preparation against rabies, you can see antibodies are produced against the sheep brain antigens. They can act on the human brain. So because of sharing of this antigen, these antigens, they are called as organ-specific antigens. Next is heterophile specificity. Some related antigens are present in tissues of more than one species. For example, Posman antigen. Another example is in well-Felix reaction, what we use, protease antigens, they are used to diagnose infections caused by rickettsia. In Paul Bunnell test, you can use sheep RBCs to detect infection caused by Epstein-Barr virus. Because of sharing of antigen, we can use human blood group O RBCs to detect infections caused by mycoplasma. All these are the examples of heterophile antigens. Some organisms, they produce superantigens. What are superantigens? Antigens that react with the antigen presenting cell and T lymphocyte in non-specific manner. And in this case, an interaction occurs between superantigens with MHC class II molecule on antigen presenting cells and variable domains of T lymphocytes, leading to non-specific stimulus of large number of T lymphocytes, leading to large quantities of release of cytokines. These superantigens can be found in Staphylococci, Streptococci, Mycobacterium tuberculosis, bacteria or HIV, rabies, and Epstein-Barr virus. Dendritic cells, macrophages, and B lymphocytes are the antigen-presenting cells. Usually, when antigens enters the body, they are dependent on the T helper cells, which will stimulate B lymphocyte to produce antibodies. But there are some antigens where the action is independent of the T lymphocytes. These are called as T cell independent antigens. We can differentiate these antigens. Antigen processing is required for T cell dependent antigens. It is not required for T cell independent antigens. T cell dependent antigens, they are easily degradable. There is immunological memory present. There is no complement activation or polyclonal activation, and all types of immunoglobulin like IgM, IgG, IgA, and IgE are produced by T cell dependent antigens. So we can compare these antigens. So what is primary function of immune system? It is to recognize and eliminate foreign cells. The primary function is to eliminate foreign cells. So when we apply a graft is rejected. So what are different types of graft? When a tissue is applied from one side to another side of the same individual, it is called as autograft. So within an individual. When it is transplanted between identical twins, it is called as isograft. When the tissue is grafted from between two non-identical members of the same species, it is called as allograft. And when the graft is in between two different species, it is called as xenograft. So usually allograft rejection occurs because of HLA antigens. What are these HLA antigens? Gorer in 1930 identified allograft rejection in mice. He observed blood group antigens in mice, and there are two types of antigen. He found that antigen 1 is common for all mice, whereas antigen 2 is present in some of the members of mice. And this antigen 2 is coded by a cluster of gene called as H2 complex in mice, which is responsible for rejection of graft, allograft in mice. Similarly, genes, they are identified which are responsible for allograft rejection in humans. They are called as HLA complex. So major histocompatibility complex in man, it is called as human leukocyte antigen complex, or HLA complex, that includes genes which code for the HLA antigens. For transplantation, it is necessary to cross match or matching is done between blood group antigens of the donor and recipient as well as the HLA antigens. These HLA antigens or human leukocyte antigens, they are present on the leukocytes. They are responsible for allograft rejection in man. These antigens, they are coded by the genes. Genes which code for these HLA antigens, they are situated or located in short arm of a chromosome 6. They consist of 3 clusters of genes, class 1, class 2 and class 3. Class 1 cluster, it includes HLA A, B and C loci. Class 2, they include DR, DP and DQ region, and class 3, it includes complement components, factor B and tumor necrosis factor. This is the short arm of chromosome 6 where this HLA complex is located. It consists of clusters of gene. Class 1 region includes HLA B and ABC. Class 2, it includes DP, DQ and DR. And class 3, it includes complement components, tumor necrosis factor. In all species of animals, genes which are present on this HLA complex, they code for the proteins, which are called as class 1, class 2 and class 3 proteins. Class 1 proteins, they are responsible for histocompatibility and acceptance or rejection of allograft. Class 2 proteins, they regulate immune response, and class 3 proteins, they are complement components. Position where a particular gene is located on chromosome is called as locus, and these genes they are present in several alternative forms, and they are called as multi-allelic. So each allele will code for a distinct gene. So there are 24 alleles at the region HLA-A and 50 at HLA-B region. So according to WHO, how these alleles, their nomenclature of HLA system, they are labeled by locus followed by corresponding antigen. For example, for HLA-A1, A is locus followed by number 1. HLA-DR7, DR is locus followed by antigen 7. Now this difference between the class 1 and class 2 molecule. This class 1 molecule, it consists of a heavy alpha chain made up of 3 globular domains, alpha 1, alpha 2 and alpha 3, and a small beta chain. There is a transmembrane terminus which enters into the cytoplasm. This alpha 1 and alpha 2, they are folded to form a groove to which this processed antigen will bind. In class 2 molecules, there are 2 chains. Alpha chains consist of alpha 1 and alpha 2 domains, and beta consists of beta 1 and beta 2 domains. So proximal domain is constant region, and distal domain is responsible. It is a variable domain, and a cleft is formed between this alpha 1 and beta 1, where the processed antigen will attach. These class 1 antigens, they are present on all nucleated cells. They are responsible for rejection of graft and cell-mediated lysis. Cytotoxic lymphocytes, which are called as CD8 lymphocyte, they will recognize MHC class 1 antigen. And class 2, they are distributed on macrophages, monocytes, activated T lymphocytes and B lymphocytes. They are responsible for graft versus host reaction and mixed lymphocyte reaction. You will learn this in subsequent lecture, what is graft versus host reaction. Class 3 molecule include complement component like C2C4, from property factor B, and tumor necrosis factor. So primary aim of this major histocompatibility complex is to produce defense against organisms, microorganisms, not against the graft. For transplantation, it is necessary to type antigens of donor and recipient. And for HLA typing, class 1 antigens can be typed using anti-sera from multiparous women or persons who have received multiple blood transfusion, or from individuals who are repeatedly skin grafted. But nowadays, with availability of monoclonal antibodies, this anti-sera, they are not used. So monoclonal antibodies are used for HLA typing of class 1 antigens. Method used is called as microcytotoxicity, where the lymphocytes, they are mixed with the standard anti-sera consisting of antibody in presence of complement. And then it is incubated. So cytotoxicity lymphocyte mixed with antibody in presence of complement and incubated, and after incubation it is mixed with trypan blue. Trypan blue will stain dead cells. So in this micro lymphocytotoxicity, these lymphocytes mixed with antibodies, complement, and trypan blue. After incubation, dead cells, they are stained by the trypan blue. So we can type these lymphocytes using these stains. For typing of class 2 antigens, HLA-D region antigens, they are identified by mixed lymphocytic reaction. In this test, donor lymphocytes, they are irradiated. They are mixed with the recipient lymphocytes in presence of radioactive DNA precursor. If the donor antigen is foreign, recipient cells, they are stimulated and lymphocyte replicate, and their DNA is incorporated in a radioactive precursor. Amount of radioactivity is measured. So you can see in this slide, donor cells, they are irradiated, mixed with the recipient cells. If the recipient cells, they are lacking these antigens, they are proliferated and they get incorporated by the radioactive substance. So radioactivity is measured. When the donor cells and recipient cells, they share antigens, there is no reaction. So this reaction is used to type class 2 antigens. HLA typing indications for HLA typing are tissue typing prior to transplantation or for paternity determination. We can associate some diseases with HLA types. For example, HLA-B27 with ankylosing spondylitis, HLA-DR4 with rheumatic arthritis, and HLA-DR3 with many autoimmune diseases. And lastly, about MHC restriction. T cell or T lymphocyte, they will respond to processed antigen only when it is presented along with the self-MHC molecule. That means CD8 lymphocytes will respond only when antigen presenting cell presents antigen with class 1 molecule. CD4 lymphocyte, they will respond only when processed antigen is presented along with the class 2 molecule. This is a cartoon which indicates how the MHC restriction occurs. When the antigen enters into antigen presenting cell, it is degraded. So processing of antigen occurs, peptides, they are released, and after processing, it is presented on the surface of antigen presenting cell. So it is where if it is presented along with the self-MHC type, then only CD8 lymphocyte will react to these antigens. So for reaction of CD8 lymphocyte to the antigen, antigen should be presented along with the MHC class 1 molecule. This is about MHC restriction. T lymphocyte can kill virally infected only when T lymphocytes and target cell, they are of same MHC type. So for CD8 cells to react, antigen should be processed along with the class 1 molecule, and for CD4 lymphocyte, it should be presented along with the class 2 molecule. This is called as MHC restriction.